Okay. Okay, Assalamualaikum. So today we have a seventh day of lecture, and Dr. Professor Yong is here. So let's over to him. Okay, thank you.
Okay, so we can start our lecture. So actually, the whole lecture series is called Applied Biomedical AI. So our main focus should be on the application of AI to biomedical tasks. So today is the first lecture we actually zoom into biomedical applications. Specifically, we look at the examples of how machine learning methods are applied to the current studies, particularly those published in major medical and biological journals in recent years. So first, we look at the application of disease diagnosis, particularly, so there are some molecular techniques that people can use to try to detect, try to diagnose the disease based on some measurement. One particular measurement is the methylation state of the DNA of the patients. So essentially, we can draw DNA samples, the samples from either from the some suspected disease tissues or from simply from the serum from the blood. So based on the extracted samples, for instance, the DNA samples, we can measure whether or not a particular DNA chain is actually modified by this so-called methylation process. That means that in the biological system, we typically have our biomolecules modified by adding some kind of molecular fragments to the micromolecules. Specifically here, the micromolecule we are talking about is DNA. So the DNA nucleotides can be modified, chemically modified by adding some chemical components to the DNA chain. So this process is called, so one for this process is called the methylation process. So anyway, all we need to know is that if a DNA chain is modified by this methylation process, understandably, if we do a fluorescence analysis, and we can detect some changes in the molecular signal, in the fluorescent signal of the molecules. So this is so these signals are the ones we can explore. We can draw certain signals as some kind of biomarker associated with the disease. So if we can find several such fluorescent signals, and we may utilize those signals, the presence, the strength, the brightness, or the colorness of the signals as some kind of features. So those features could be input into a machine learning method. So that's how this particular article published in 2018 actually explore. So it is published in Nature. So they look at a very hard problem in the diagnosis of CNS tumors, brain tumors. So they can draw certain the samples from the brain, but the question is because of the the nature of the samples, it is very difficult to actually determine the cancer state, particularly for those with very low density of tumor cells. And because we prefer to predict as early as possible, particularly after the operation, you don't want the redevelopment of the tumors, for instance. Or if we suspect that there's a brain tumor, we want to diagnose that as early as possible. So this is one of the approaches that people has tried. So essentially use this demethylation state. So in this particular article, they explored the random forest model. So remember that in random forest, we need to first to extract some features out of all possible features, and also we need to so essentially extract a fraction of the patients to train the model because we have the freedom of multiple fractions of patients and multiple group of features. We can develop a forest of many decision trees. So those forests actually give us the predictive power for diagnosis purposes. So here is just an outline of the procedures. So understandably, the strategy or the methodology is quite straightforward. You just draw some features, specifically methylation features, and you just select a fraction of the patients or whatever samples you can collect, and then you develop the models. But of course, practically, there are some technical issues. So that's why we highly recommend you to try out yourself, because if we keep talking here, it's just like we we learn swimming from a book. So that's always not the way to do that. So the way to do AI is to practice it. And so actually today we are going to have another practical session and help you to go through some software. Once you have the software, you can simply grab the training data. So typically, like this article published in Nature, like this one, in Nature or in Nature Medicine, typically they provide you with the training data either as a supplementary material, or they actually post it on some website, or else you can simply download some kind of data from GEO database, from the TCGA database, for instance. Those databases that I already mentioned in our the data data lecture section. Okay. So now we look at the second example. In this case, machine learning method is was actually or has been applied to the diagnosis based on the metagenomic data of the stool samples. Metagenomic data of the microbiota essentially in the gut microbiota actually has lots of applications. So some of the applications are listed on the right-hand side. Because we know that the gut microbiota is connected to many of our tissues, like through such a thing as a gut-brain axis, gut-lung axis, and so on and so forth. So because of this linkage, gut microbiota is a very good source to explore for diagnosis purposes. So typically we measure essentially we measure the abundance of the microbes in the microbiota. So there are multiple microbes. So obviously we have multiple the abundance of multiple microbes. So for this complicated measurement, the technology applied to is metagenomics approach. Essentially, we simply extract all possible DNA fragments from the stool samples. So it contains the DNAs of many microbes. And then there's this technique to try to figure out which microbe a particular fragment come from. And then based on the number of fragments, we can actually derive the abundance of the species, the microbes, the species of the microbe. And so the abundance of the species is somehow linked to disease state. Understandably, in a certain disease state, certain microbes, they have abnormal abundance. So this is the scientific principle why we can use machine learning method to actually learn about the ability to diagnose the disease based on the metagenomic data of the stools. Okay, so here is the example published last year in Nature Medicine. So its task is to diagnose inflammatory bowel disease based on the microbiota, the metagenomic data of microbiota. Again, random forest method was employed for developing this diagnosis machine learning methods. So talking about diagnosis, typically there are two main approaches in diagnosing a disease. One approach is to select one or a few, or at least not too many biomarkers, and based on the abundance of those biomarkers to figure out whether or not or to diagnose a particular disease. So the well-known example is just like HER2 positive breast cancer. We know that HER2 is obviously a biomarker for the diagnosis of HER2 positive breast cancer. And there are some other diseases that have this kind of distinguished biomarkers. But what about a complex disease that may or may not have a so distinguished biomarker? So chances are those diseases, they are somehow regulated, or there are some group of so-called biomarkers, either they are genes or they are microbes or whatever, they actually change in response to the disease process. So their state, the abundance of those biomarkers could also be used for diagnosis purposes. But the abundance may actually have different combination of abundance actually corresponds to the same type of disease state that is possible. So in that case, obviously, a simple biomarker approach, explicit biomarker approach, no longer works for this complex relationship because you need to have so many if, just like the first example we illustrated. So in this case, in this second case, a machine learning model is very helpful because it can actually deal with this kind of complex combination type of things that correspond to the same class, disease class. So anyway, so this is how they actually develop this kind of random forest model for diagnosis of this inflammatory bowel disease, this kind of process. Again, the important thing is the the selection of the samples from the cohorts, and also the selection of the biomarkers. That means the specific species, the microbes, and using the abundance of the microbe as the features for developing the random forest models. So this is how they actually have done this. So on the right-hand side, lower right-hand side, you see they listed several species. Unfortunately, some of these species, they don't truly disclose the real name because they want to keep the commercial power, the applicability, commercial potential to their own advantage. So this is somehow the difficult part when we try to further develop this kind of approach based on these so-called scientific publications. Frequently, you encounter this problem. The precious information is somehow partially blocked because they don't want to really, particularly those magic species. These species are very, very useful, very, very important, not only for the diagnosis purposes, but also for the mechanism study purposes. But unfortunately, it may or may not be disclosed. So this is the problem. So we look at the third example. So in this case, the genome of the disease, for instance, the human genome for the human disease, like the cancer, the tumor cells of the genome of the tumor cells, because maybe the tumor may be caused by the mutation. So obviously the genetic sequence can give us some important features for diagnosis purposes. For genomes, I think you all understand that, right? So on the right-hand side, we have a biological species, our human, ourselves, and then our human, any biological entity is composed of cell as the smallest unit of life, right? So inside the cell, there are some structures, particularly the cell nucleus, and inside it contains this very important uh, uh, system called chromosome. So chromosome is composed of DNA and chromatin proteins wound into this kind of structures. If you uh, uh, look closely to the DNA, you see the double helix structure, and then you see the sequence of the DNA. Okay, so some sequence, particularly some mutant mutations, actually uh, uh, is the driver of the cancer or the driver for the spread of the cancer. We could zoom in on certain sections or certain parts of the genome and then use the mutation states of those parts as the features for diagnosis purposes. So here is an example of the use of genomic or genetic features uh, and use those as features and employ a decision tree machine learning method to develop a diagnosis model for the prediction of the metastasis of cancer. So the next example is a typical clinical metabolic test. Typically, the blood samples is drawn from a person and then the metabolic some kind of metabolic type of measurement was done. It could be simply the metabolomic measurement, or it is simply a fixed measurement of a certain a smaller group of metabolites. So it depends on the specific situation. So by measuring the metabolic states of the blood samples, and we could be able to diagnose different disease, specifically in this case, the diabetes, type 2 diabetes disease. Okay. So for this particular case, they actually look into some subsets of metabolites, and then based on the profiles of those metabolites, they develop machine learning models to diagnose the not only whether or not a person has the type 2 diabetes, but also want to predict the subtypes of the diabetes. We know that the different subtype of disease may require a different treatment. So it is important to actually classify patients into different subgroups, and each subgroup is with a optimized treatment that typically differ from the other subtype groups. So this is the approach. This is a paper also published last year, but you can see that it has been under review for almost so actually over 2 years. So you can see how difficult it is to publish a biomedical AI model in this kind of prestigious journal. In this case, it is Nature Biomedical Engineering, because you need a lot of the examination, analysis, and the evaluation by the reviewers, and then you need to make an improvement, or you make to clarify why you want to do this, why you want to do that. In this particular case, you notice that they employ not a specific machine learning method, but rather a group of machine learning methods, including support vector machines. Even for support vector machines, they have a different version of support vector machines. And also they employ logistic regression, as well as that famous random forest. So this example shows that sometimes there's no, of course, there's no general rule that one machine learning method is always better than others. Sometimes you may find that you will need to employ the power of multiple machine learning methods. So this is actually a well-established approach in computational science. It is called consensus modeling. So if you have done molecular docking, and particularly those earlier publications, when they try to establish which molecular docking method are better than other, because there are so many molecular docking softwares. And you'll notice that some publications actually advocate the employment of consensus modeling. So consensus modeling means that you use not just one model, one method, but rather use several methods, and each method is considered as one judge, just like some sports events like gymnastics, those kind of things, that we need multiple judges because we don't want the bias of one particular judge. If you have multiple judges, So people found out that even though it becomes complicated, but somehow the consensus decision seems to seems to outperform uh, the decision made by just one judge, particularly for those hard to distinguish problems like gymnastics. How do you deem this? Yeah, this person actually performs better than the other person because sometimes it's up to personal preference, impression, so many different factors, right? So you want to get away from the bias for those complex situations. The best solution is the use of multiple charges. So here is the same thing. So the multiple charges have been used in this specific paper. So now you can sense why they need 2 years for the review of this paper, because you can imagine the problem may be very hard to solve. So they have to go several rounds to figure out the best approach, the best clarification. So in the end, the multiple machine learning models seems to give the good performance. Okay. The next example is a single cell technology. If you are active in molecular biomedicine in those fields, and you notice that single cell technology are popular ones. It is in contrast to the bulk the transcriptomic studies of disease samples, the patients and the animals, those kind of studies. In the bulk transcriptomic studies, you simply extract the tissue samples, and then you extract the RNA, and then to do RNA-seq, those kind of studies. But for a single cell technology, we do at least one step further. So we still withdraw the sample from the patient, for instance, the tissue sample, but we need to separate the cell types, different cell types from one another, and then we zoom in on a particular single cell type to study the transcriptomic to do the transcriptomic study. So you can see this actually illustrated the procedure. So we have the patients, and then we have the samples from the tissues, and then we do the single cell culture to extract a single cell to separate, or rather separate the single cell, and then we do the CD amplification, and then we do this kind of single cell transcriptomic measurements. So this is the process. So in this particular paper published in Nature Genetics, and the single cell data was employed to actually identify certain genetic regulators for a particular health problem. So in this health problem, and a sample was placed in an environmentally stressful condition. Under those conditions, we can expect the sample should have some uh, re response. In this particular case, this is a glioma. So essentially, it is a glioma, kind of a cancer problem. So we can imagine that the environmental factor can make the cancer process even worse, right? We want to study that. So in this paper, they actually draw the single cell sample, and then they first employed the unsupervised clustering method to try to separate the samples, separate the features, and it into different subgroups. And remember that we talked about the heat map, right? So in the heat map, we can have samples grouped based on the in the sample space, or the genes grouped in the genetic space, right? So if we can do that, we can actually select those genes that are grouped together to be some kind of uh, regulators or or markers or regulator, because uh, those regulators that are associated with the disease tend to have certain common behavior. For instance, they must be active during the metastasis, during some kind of process, right? So depending on what process you measure, okay. And those more active ones, they typically they are elevated in expression level, and then they should be clustered close to each other in our unsupervised learning method. That's why a unsupervised learning method was employed here. I think one of you has asked a very good question: how do we visualize the clustering method, right? So in this particular paper, they use the dimensionality reduction method. It is called MDS, and then to reduce the dimensionality from the high dimensional multiple gene space into a two-dimensional space, whereby you can see how the samples are separated in different colors. So those samples are the single gene, single cell samples. Okay. Next, we look at we already mentioned about the transcriptome. Now we look at the transcriptome. So transcription process is illustrated in this essentially in this animation. So we have a double-stranded DNA. So they are in the closed, so the normal form, right? So in order to read the genetic code of a particular gene, or in another word, if we want to transcribe the genetic code of one gene, we have to separate the double helix. So there are some enzymes called RNA polymerase. RNA polymerase can actually help us to do that. Not only it separates the two strands, it also needs to transcribe to actually translate the genetic, the DNA sequence into a RNA sequence called the messenger RNA. So this is kind of a process. Okay. So here is the example of how transcriptomic data was utilized for biomarker discovery for a certain kind of health problem listed here. So it is published in 2020 in PNAS. In this case, we have the sampled the sampled drawn from the patient, and then the transcriptome was measured by DNA microarray. Of course, there are some other laboratory measurements measuring the blood sample measurement. But based on the transcriptomic data of microarrays, we can, so they actually, the authors, they applied unsupervised clustering method to cluster the data, and so that you can actually draw certain features that behave abnormally. They behave in a similar way abnormally similar way, abnormal way in the disease state. So those features are analyzed for the selection of the biomarkers. So this is the process. After the selection of the biomarkers, they can actually do this lab test. In the lower part you can see they can do this real-time PCR to actually focus on those biomarkers and to see whether or not they can really distinguish the disease state. So this is the work done by this group of people. Okay. Today we just give you several examples, and each one covering a different type of molecular features, different type of genetic test, and also I try to give you examples in different that employ different machine learning methods. Okay. I think all these examples they tend to be strategically or the methodology in methodological methodological aspects, they actually are very understandable, very similar. But of course, each specific example is quite non-trivial because the way how you select it's too easy to say select some features, but it is very hard to really do it in a good way. In a satisfactory way. So because you want your model to be a high performance one, right? So I think the only way to learn that is by practice rather than by looking into more and more examples. I don't think that's productive. So that's why today mm, we are going to have another practical session. So before the practical session, maybe it's time for answering your questions. Do you have any questions?
Okay, thank you so much, Professor. Let me ask them from them any questions. So, in our audience, do you have any question from Professor? No? Okay, sir, let me check it from a chat box.
Okay.
Sir, the one question it is by Kash Mughal, and he's saying why addition of methyl group related to diseases?
Because you know, so we know that a disease process or physiological process is actually preceded or driven by certain proteins. The proteins, they are because we need some workers to actually perform, to do something, right? So the workers are proteins. But sometimes, so the proteins actually perform these works by molecular interactions, by catalyzing chemical reactions, and so on and so forth. In order to do this, and we need different workers. But we don't want our genome, our DNA to actually explode by including so many different proteins. So how do we generate more workers, more different type of workers without significantly expanding our genome? So the only way is that we use chemical modification. Without a particular modification, it is protein 1, protein A. With modification, it may be protein B, protein C. So that's why we have all these kind of methyl or other type of modifications. And we can also not only on the regulation of DNA, but also on the protein itself. And so the regulation of DNA means that we can actually manipulate which part of the DNA is transcribed. For instance, we can transcribe one short section from region A and then another short section from region B and combine them into a single protein, a hybridized protein. So that's one strategy. There are many these kind of tricks our biological system can do. All these tricks, and we can also directly modify a protein like a phosphorylation of a kinase, right, to make the protein in different perform differently in different states and so on and so forth. So all these actually require this kind of chemical modification. So that's my answer. Okay.
Okay, so thank you, Professor. We have another question that is by Muhammad Hassan, and he's saying, can we use PCA on small data set with a very few samples?
That's a good question. So again, as I mentioned that if you want to develop a good model, you have to consider the marriage or the linkage between AI, between computational method and the biotechnology, the biology side. To answer your question, you can ask yourself, are these few samples representative of all the patients you want to diagnose? If yes, then it is possible. If no, you know these samples are not enough. So it is questionable. For instance, if you want to consider is a non-small cell lung cancer, for instance, for this cancer, how many patients are minimum to cover all possible subtypes, all possible individual variations of the non-small cell lung cancer? I imagine there should be a minimum number. If you don't reach that number, or if the diversity is not satisfied, we know there's a deficiency of the model. So the first question is in biology, in medicine, rather than in AI. And then the second question is, okay, suppose these few samples are good enough. How do you develop a good model to distinguish them or to diagnose them, right? So that's the second question. So in this question, on the AI side, we need to ask, can the patients and the non-patients can they be completely separable, completely distinguished by AI? For instance, if we can't tell the difference between a patient in early stage and a normal person, so if we can't separate them, we cannot predict the disease, right? So I can tell you if the sample, so that's why we need to, well, that's why the large models, the large language models in language and the large models in medicine, in chemistry and in biology, they are so important because those large models may provide the basis for a clear separation of all the samples. Once the samples are separable, then you can ask whether or not you can place the sample in the right place. That means a disease and a non-disease. So this is up to the training. I can tell you that based on the large language model, because those language samples, they are perfectly separable. So they test that with a few samples, they can train a sensible AI for the classification task. So that's based on the language example. So I can imagine those phenomena should also be applicable in the case of medical AI. But unfortunately, we do not really have a large language model equivalent large model for genetic samples and also for biological, meaning the protein samples. We do not have as good a model yet, but hopefully through the effort, collective effort, particularly some of you, I think it is possible in the future. Okay, that's my answer.
Okay, thank you, Professor. It's the last question by Muhammad Asfandyar Khatak, and he's saying कि if we cannot visualize data even with dimensionality reduction, how do we know if we have selected an appropriate k number for clustering?
That's a good question. So some computational scientists designed some algorithms because of the nature of this lecture, so I did not include those methodology into the lecture. So those methods, they are the methods better than nothing, but they actually do not work very well. So they may be helpful, but definitely they are not really deemed as a good model. So that's the reason why I didn't even bother to include that. But I can tell you, computational science, they tried. So essentially, the strategy is they look at the size, the density of the class, because you can, so if the class, for instance, you can imagine if the class occupies two large areas with a very low density, you can even imagine yourself, they must be placed into several classes rather than a single class. If a class is too densely packed and not too far away from its neighbors, you can imagine that why not join them into a single class? So this is the consideration those computational scientists they thought they try to develop those methods, but they are not really popularly used. So again, I think the biological science really helps. So that boils down to the marriage of AI and BT, biotechnology. You need to really think from biological science, how many, so essentially, you know the samples, you can randomly select the samples, you know the gene, you can randomly select the gene, and you can judge whether those samples or those genes, are they really should be in the same class or they really should be in different classes. So this is called the biological knowledge, okay? Biological knowledge should give you more, actually, a better hint about the whether or not the clusters are reasonable. So this is essentially my suggestion. Okay.
Okay, thank you so much, Professor. And I think then we are over from this to lecture, and we are giving this
Okay.
session so Dr. Wang.
Okay, we'll give it to
Yeah,
Dr.
today
Wang.
the practical
Okay.
session. Yes. Okay, thank you.
Okay, thank you.
Yeah, can you hear me?
Yes, I'm hearing you.
Okay, I will show my desktop. Okay, hello everyone. Good afternoon, and my name is Wang Likun. I'm a associate professor, assistant in Ningbo University. So today I will give you a practical session, and wait a minute. Okay. Uh, so the first thing to use machine learning, we should install Anaconda. Anaconda it's a software, and there are many videos to teach us how to install Anaconda. And I will I found a good video, and I will show you.
Dr. Wang, can you hear me?
Yes, I can hear you.
Okay, so is there any video or sorry, is there any audio of this video? Can you?
Video.
Yes, okay. So can you pause the video and explain the steps? Because so that our audience understand what is happening.
Uh, you and then the second step, we need to install sklearn.
Dr. Wang, can you please
Yeah.
explain? Yeah, can you please explain what are the what was what was done in the video number 1? What were the steps?
The purpose of video 1 is to install Anaconda.
Okay,
And then okay, yeah, if you install Anaconda, then you need to install scikit sklearn. So when we finish install scikit-learn, then we can use many machine learning algorithm. And here is the example, k nearest neighbors. Here it's the speaker use the AROS dataset. So if you don't understand this algorithm, I put the video link in the chat box, so you can watch the video after class. Thank you.
So the links have been posted by Dr. Wang, and I'm going to copy them, and we'll be sharing that with all the participants. I would also request Dr. Wang, are you here with us? Dr. Wang?
Yes, I'm here.
Okay, so Dr. Wang, for the next practical session, I request you to please, because we were not able to, listen to the video that you were playing. So please check if there is a sound with the video. That's one thing. And also please explain in brief steps the work that you were doing, you were trying to show through the videos.
Okay.
Because right now we were just looking at the video and trying to understand what was happening. So I request you that next time, please do a little bit of explaining for our participants.
Okay, actually the video has sounds, but I don't know why you can't hear.
Okay, maybe next time we will see. Otherwise, you can always send me a link through WeChat, and we will download these videos at our end.
Okay.
Okay, thank you very much, Professor Wang, for joining, and we hope to see you in the next session, inshallah. And just for all our participants who are present here in person and online, this is the first session. The second session will begin on 11th of August. So you are requested to please join online or in person from 11th of August. And similarly, we hope to see Dr. Wang and Dr. Chen with us from August, inshallah. Thank you very much, Professor Chen and Dr. Chen, for your time, and bye-bye to you all. Nasir, can you call Mumtaz?